Okay, John, take it away.
All right. Hello, everybody. I'm John Schloendorn, the CEO of Gene and Cell Technologies, an up-and-coming regenerative medicine company. Before this company, I worked at ImmunePath, where we derived immune cells from embryonic stem cells and saved a lot of mouses' lives. But ImmunePath failed because people did not want to give us money to save human lives. Mice are more important. Yeah, so I started this new thing, which is an integrated regenerative medicine company. I have no research going in the field of Alzheimer's disease, so I declare no risk of
workout. What? 24-hour fitness. Yeah. Which? How many are downtown Oakland? They're fine. No, all over the place. No, I mean, how many in Oakland are there? Oh, just one is like the sketchy part of Oakland. It's like near the group now.
Sorry, guys. Someone is not getting through there. One person at a time.
Can you see us, or?
I can't see us. I don't have a camera. Sorry, but.
Hey, John. This is Ryan and the counterculture team. We are here.
Hello.
You just can't see us because there's no camera.
Okay, welcome. Over here, I'm in the headquarters of Gene and Cell Technologies. I can show you around a little bit. There's our giant whiteboard for planning things, printer and 3D printer.
Hi.
This is like dishwasher, so that's the room you're in. We keep the biotech in a different room so you don't get to see any creepy stem cell projects in this session. Like I said, we have no research going on in Alzheimer's disease, so no conflict of interest with the current paper. Let's go ahead. Yeah, so last time we had a lively discussion about how we could possibly target protein aggregates in protein aggregation disease, such as Alzheimer's disease. And people came up with these nagging questions like, wait a minute, is that desirable? Is Alzheimer's disease really a protein aggregation disease? Everybody's heard about beta amyloid hypothesis, Alzheimer's disease. And so I remember reading a paper that seemed to refuse that, but I forgot most of it. So I was asked to go back and dig that paper up, and that's what you've hopefully all read. So what happened in this present paper, let me bring that up here, is that Elan Pharmaceuticals ran a study with a vaccine against beta amyloid as early as 2000. And their goal was to program the immune system with a vaccine to clear beta amyloid from the brains of Alzheimer's disease patients and see if they improve and if they can make billions and billions of dollars by curing this very, very major disease in that way. And people were very excited about this at the time. The beta amyloid hypothesis was really important. It turned out shortly thereafter that that vaccine failed, had no efficacy, had some serious side effects, no obvious improvements in patients, and has been shut down since. Now, the current paper is a group of academics picking up these patients 6 years later to figure out what actually happened in the brains. So the question is, did the vaccine do its job? Did it clear beta amyloid and then not result in cognitive improvement? Or did the vaccine simply fail to work and therefore the beta amyloid hypothesis may still stand? And that's where I'd like the group's input, and I like to tap all of your critical thinking to figure out if this paper can really confirm or refute the beta amyloid hypothesis, or if we still don't know. So to the heart of the matter is I'd like you all to look at figure 2. These are slices of the brain. Do you all have figure 2 up there? Slices of the brain from patients that received the vaccine. And the black spots in figure 2 are the beta amyloid deposits, right? And you can see that some patients responded really well to the vaccine, supposedly clearing all the beta amyloid. Others didn't do so well. But what's written into these pictures, and that's normal for a vaccine, and sometimes they work better, sometimes they work less well, right? So this is what we expect to see for a working vaccine. But we have to take this apart and make sure that it's real. And what's also written in figure 2 is MMSE. That's a cognitive test score. And you can see all these patients have MMSE 0, meaning they're about as responsive as a tomato when they die. And that includes patients with very clear removal of beta amyloid. And that's why this paper claims beta amyloid is unrelated to dementia. The patient's progress, just like the controls, and beta amyloid is at most a bystander in Alzheimer's disease.
Hello, can I hear you? Okay. Quick question. You said some of the vaccine worked for some of these patients because they had less or no beta amyloid plaques, but is there any way that you can actually do a brain biopsy while they're still alive beforehand, or is this just all post-mortem and just conjecture that they had beta amyloid plaques because of their symptoms? Is it just purely based on symptomology?
Right, good question. So,
is that what they use?
No, I'm saying they did, but that would be a good.
So, or as a control, did they take brain? Oh, they did. So they're comparing it basically to a cohort that was not had Alzheimer's, correct? Or and that had.
Well, no, the cohort had Alzheimer's and wasn't vaccinated.
And wasn't vaccinated. Okay.
Right. People without Alzheimer's aren't supposed to have plaques, right? Because Alzheimer's is defined as plaque dementia.
So people that do have Alzheimer's, 100% they have these plaques. It's like not known, like nobody spontaneously just has all the symptoms of Alzheimer's.
A lot of questions there. Let's tackle them one at a time. So,
the controls.
Alzheimer's disease is defined as dementia with plaques, right? Hey guys, you still there? Yeah, okay, yeah. So Alzheimer's disease in particular is dementia with plaques. Now, it is certainly possible to have dementia without plaques, and it's also possible to have plaques without dementia. And these are some of the results that make the whole amyloid hypothesis seem questionable. But those are different papers, and they're really interesting too.
So in paper, they also mention actually old people who have the similar level of plaques in their brain, but they didn't have dementia. To your question.
Yeah.
Yeah.
So there are old people that when they do postmortem autopsy, they find amyloid plaques like this, but they don't never had any signs of dementia.
Yeah.
Right. But to really answer that question, we would have to have a separate session where we take that paper apart, right? Because there's a lot of stuff written in papers, and you know, the point of this exercise is to really use critical thinking and focus on this one thing. So what we have in our patients, that was your first question really, is do we have beta amyloid images from these patients from before they were vaccinated? Can we image them in vivo, right? And the answer is, back then we could not. So these patients are diagnosed as probable Alzheimer's cases because we couldn't image the plaques then. And back then, it was possible to diagnose Alzheimer's disease only post-mortem because only then could you see the plaques. So before that, they were probable Alzheimer's and they were diagnosed with dementia. So we unfortunately, and that's right at the heart of one of the big weak spots of that study, we do not have images of these brains from before. And that would be great, and that would allow us to come up with a much better answer than it currently does. Yeah, so totally, you spot on question. Now today, we have methods that allow us to image beta amyloid plaques live, and that would be exciting to repeat. But today, everybody and their uncle dropped their Alzheimer drugs, so we don't get living patients getting a promising drug like that because that field is, you know, closing down in the wake of studies like this. So that, did that answer your questions?
Yeah.
Cool. Okay. Right. So, I mean, now what I would like you all to do is go through the study and try to poke any holes in the claims made by Figure 2 and made in the conclusion section to see if is there any way we can defeat Figure 2? Is there any way we can show that Figure 2 doesn't mean what it seems to mean? Right? So I'd like to go with you through this study pretty much one chapter at a time, one claim at a time, and nail this all down. So first to the group, does anybody want to improvise? Does anybody have a view? Or should I start? Does anybody have something on their mind, or should I just start guiding you through chapter by chapter? I'd like you to be lively and participate. So if anything comes to mind, please go ahead. What do you want to talk about relating to defeat Figure 2?
Wait, can I ask a question on some background information?
Go ahead, by all means.
Okay, so amyloid plaques, I know they're made up of, there are some like known proteins that make up these plaques, but I'm also assuming that there are maybe some other protein aggregates or other proteins that may be recruited to these plaques that are potentially not known, or the lasting damage caused by these the types of cellular damage that is caused by these plaques. Like, is that fully understood or known? So I guess before I understand every aspect of this and other things that could have been tested for, especially in the like histology portion, I was wondering like what are some other, if there are other known markers for the effects of these amyloid plaques?
So yes, definitely there are all kinds of proteins and all kinds of even broader materials in there, right? Mm-hmm. So what seems to be the case when you look at how the plaques progress through the brain, right? What seems to be the case is that the plaques get seeded with beta amyloid peptide aggregates. But then when you aggregate a random, like, semi-crystal like that in the brain, of course you get other molecules attracted to that, like cells that examine what is going on here, all kinds of immune cells, and then you get metals that get soaked up into that, and you get other proteins getting encased in that. Right. So that's why they call it the beta amyloid cascade hypothesis, that says this thing seems to originate with beta amyloid and then draw some other damaged components in there and encase them or attract them in some way. So definitely that is what happens, but people largely believe that it originates with A beta. And so the question is about A beta itself. Is it really the true culprit that initiates everything? And then also the question is, are these entire plaques, are they pathogenic or are they just bystanders with all the stuff that they have in them?
Yeah, exactly.
And so what we're staining there is plaques with a dye that's supposed to be one of the compounds that's soaked up into these spongy plaques, right? That's how that dye functions. So we're probably imaging plaques at all kinds of stages of the process there, and we can't necessarily resolve how pure they are in terms of A beta with this other stuff.
Yeah, got it. Okay.
But one thing they did in the study, they only used patients with medium and severe Alzheimer's disease. They didn't use people with mild or early stages Alzheimer's disease. So amyloid plaques can still be the cause of the disease. They just can do permanent damage, and then it doesn't matter if you clean up them or not. The damage is done and the brain deteriorates by itself. Like they can be the root that set up the process, and then they're not required. So how you separate this cause from correlation, causality and correlation. I know if everybody received, I sent out another couple of papers, and one of those papers claimed that when they immunized mice with a similar serum on the really early stages of Alzheimer's, they actually got better. It actually helped. So
my corollary to your question is what's, well, I guess maybe I would have known this if I did more background reading on Alzheimer's. Is there an increased incidence of cell death in Alzheimer's brains? Is there a correlation between this increase in beta amyloid plaques and less neurons to begin with? So, these patients weren't helped, but, then again, they were late-stage Alzheimer's. Is there more neuronal death taking place?
Okay, so too many questions. I'll get the easier question first. Yes, of course. Neuronal death is a big hallmark of Alzheimer's disease. Brains can be half the size they used to be, especially the cortical regions. Okay, but the other question I want to really get from the gentleman to the right is another one of the big points of discussion that we're going to have here, right? So if I may paraphrase the question, could it be that the damage was already done when these patients received the vaccine, right? Were they late-stage patients? And could it be that maybe A beta has its causative effect early in the disease, right? So that was the case with some other published studies. And I thought originally, when I misremembered this paper, that the patients were very late patients because you're supposed to try drugs on very late patients, even early drugs than that. But in fact, it turns out that these patients were classified as mild to moderate Alzheimer's disease. So these were fairly early. And the figure that really answers your question as best as we can is figure, let's see, 5, I believe. Yes, if you could all go down to figure 5 and progress from their mild and moderate diagnosis to severe dementia, right? So that is just a declining score in that cognitive test that they run on the patients. And there you can really see that how the patients, the control patients, placebo patients deteriorate at exactly the same rate as the treated patients over a fairly long period, that is years, right? So that is throughout much of the progression of the disease. We see no differences in the rate of deterioration.
I have a somewhat related question to Dennis's. Since the plaques are kind of the end result of this cascade, as you call it, is it a possibility that the intermediary phase, like the oligomers, are actually the problematic or toxic part of this? So actually doing this sort of process could even be worse because it would be breaking apart the plaques back to that sort of intermediary phase, which might be the truly toxic part?
Right. So that's another important question we have. So let's maybe finish this. So I just wanted to ask about this progression chart, right? We will get back to that question in a moment, but it's a separate question. So let's finish the first one first. So if you look at figure 5, does figure 5 leave room for disagreement, right? So one issue with figure 5 is we have historic controls, the different cohorts, because none of them placebo and patients from this group.
Yeah, but how figure 5 tells us they are non-severe patients? It doesn't.
That's what tells us. The text tells us where it goes under methods, patients.
Okay.
That's where they tell us that they were classified according to MMSE scores as mild to moderate.
Oh, okay.
And in figure 5, yeah, sure, that's probability of progression to severe. So in a way, that's telling you they weren't severe before, right? And if you look at like how Alzheimer's disease develops over the years, right? You're looking at a period of like 6 years, something like that, down here in figure 5. So that is most of the progression of Alzheimer's disease that happens in that time frame.
So you
could still argue, I think, that there's a very early stage. Sorry, go ahead.
Can you explain? So 14 to 26 is mild to moderate, which is what the number will be severe and
lower than that.
What?
0 is the worst.
Oh, it's the other way around.
Yeah, you get positive points for answering questions correctly, and if you can't, then you have a lower score.
Oh, okay. So yeah, I misunderstood that. So if you go to 0, you're dead, and what the highest number is?
It's like 24, I think. Let's look this up.
It says 14 to 26, so probably 26. Oh, yeah. Okay. I misunderstood. I thought like 14 to 26 is really high number, and 0 is very low. So they took actually very mild cases. Okay, thank you.
All right. So, but still, if you technically, there is a possibility that maybe only the very seeds of the plaque are toxic at the very beginning of the disease, and then it would damage the brain so much that even removing them completely, doesn't hurt. So that gets us to the question of oligomers. Could it be that oligomers are the toxic species, right? So, and this paper doesn't allow us to really ask that question, but what we're doing is we're vaccinating against a monomer.
That was my next question.
Right. Some people have said, so there are two lines of reasoning that I can come up with. So the first one is, if we're vaccinating with a monomer and we're hitting plugs, it's kind of hard to see how we would not be hitting oligomers, right? Because the plug is already, an aggregate should be harder to hit with a monomer than an oligomer would be. And then the other one is, some people believe that the plugs are in equilibrium with the oligomers, meaning if we target the plugs, more oligomer would be converted into additional plug, right? So you'd be pulling the product out of the reaction, thereby shifting the equilibrium. But, this would take a lot more literature digging to answer. And ideally, somebody would find these frozen brains somewhere and use these specific oligomer antibodies that we now have to figure out if the oligomers were cleared in these brains. And that would be a cool study to do, but as far as I can see, it hasn't been done yet, unfortunately.
Yeah. Do you know if these self-assemble in vitro, or if they require other proteins within the cell to help start the nucleation process?
They assemble in vitro, yes.
Okay.
At least you can find conditions where they do, so they require conditions.
Okay.
I have another question related.
Virals that can form.
I have another question related to that. Although it might, deviate from this paper, I'm just curious. Do you know if there have been other studies where you've started with a base of, young, healthy organisms and infected them basically with this, and at what stage they actually exhibit dementia-like symptoms before the plaques or not? That kind of stuff. Starting from the base of healthy and inducing this.
Like injecting
injecting a prion and seeing at what stage of the cascade does symptoms exhibit.
Okay, good question. So you ask about young, healthy organisms, right? So of course not in humans.
Right, like mice or something.
In humans, the only way to do it would be to do a very long-term study where you watch for plaques to spontaneously develop from the very beginning, right? Which would mean that the investigator will die from aging before they get to publish a paper, and they don't like that. So no, in humans that hasn't been done.
Mouse models or something.
If you inject a beta, I don't know. I think this has been done with prions, like prion protein, but I'm not sure this has been done with a beta. I don't know.
That would definitively answer whether A beta has to do at all with Alzheimer's. If you do that study, the organism develops plaques from nothing and they don't exhibit dementia. That seems to be the death knell for this theory then.
Yeah, so the way the mouse models work, right? The way the mouse models work is not by injection, but by genetic overexpression of human version A beta, especially the engineered versions of A beta that mimic mutations that you can have to have accelerated Alzheimer's disease.
It's actually what you're asking.
Yeah.
They kind of injecting using DNA, synthetic biology. So
assuming there's not other.
Yeah. So they do it by overexpression, right? And what we see in these mice is that these mice do get dementia, and the vaccine fixes them. It fixes both their plaques and their dementia. That's why this vaccine looked so promising in 2000.
Is there any way to reliably detect, on average, how far along in the cascade you are? From the seeds to the oligomers to the plaques. Like, could they say at what stage did they start exhibiting dementia-like symptoms? So you know whether it's the plaques or a precursor to the plaques.
The mice, yes. So you can make mice demented with both plaques and with oligomers. What gets overexpressed is a monomer, right? And then the monomer just does what it does. It forms both oligomers and plaques.
I'm saying
so
there's no way where they've blocked the plaque formation from the get-go, but the oligomers do form and still dementia. Like I'm trying to say, is there a way that they've isolated, just mainly plaques with little oligomers and mainly oligomers with little plaques? And is there any difference there in the cognitive abilities?
I don't know. Okay. There are people who certainly believe that they have shown that, but I don't remember the details of that. But that's a good question. Yeah.
So I have another basic question. I'm sorry for interrupting, but how do we know that the measurement of the level of dementia is reliable? Because it looks like a psychological measurement. And
they said they use the same person, so like the same assessor. So at least it's,
I understand, but and of course, okay with people, but what about mice? How the hell do you compare dementia in mice with human if it's completely psychological and mice doesn't have 90% of our psychology? So how you compare those things?
I think it's like tests on how they perform in mazes and like that kind of stuff.
Yeah, so did they test people how they perform in mazes then? Because the different parts of brain are actually responsible for spatial orientation and for cognitive thinking. So probably the cognitive thinking get more impaired than the visual spatial orientation, and probably the vaccine would help, with spatial orientation, but will still leave you, with major cognitive damage. That's my really big question out of this study, because
You're saying it might have benefits in some psychological aspects.
Yeah, I think the psychological measurement is by definition is extremely unreliable measure. And my question, then the next question, is it other more measurable way to detect the Alzheimer disease, or it's all psychological effects and that's it?
Well, so yeah, I mean, good question. So I mean, if you're talking about the difference between an MMSE score of 14 versus MMSE 16, right, then I would side with you. But if you die with an MMSE of 0, you are responsive as a banana. That is very clearly a severe cognitive impairment. The test is published on Wikipedia. You can take it. It's some very simple questions, and if you're unable to answer them,
Yeah, but you see, when you do you measure psychological measurements, you need to normalize by psychology too. This study doesn't normalize, so there is a probability that people who sign up for this study have some brain dysfunction that makes them non-representative to the general population. They probably were born a little bit with dementia when they were like age 1. And that's why they sign up for the study. Everybody else, they are normal. They don't want to sign up for the study where they will dissect your brain. When you start measuring the psychological effects, you need to normalize by income, behavior, nationality, age, a lot of things. Otherwise, you can get very non-representative study, like the classic experiment with jailers. When they make students who one student jailers, another one being inmates, and they showed that people go crazy. But then another guy showed that people who sign up for that kind of studies are already predisposed to violence. The same people who sign up for the study where their brain will be dissected, they probably already a little bit dumb. No, I mean, in a simple word. So that's why I'm really pessimistic about all the psychological index measurement. I agree with you when person gets some physical signs 100%. It is Alzheimer, but how you measure the things between 26 and 14, and then between 14 and 10. Can you give more details? How it's measured? What kind of signs are you looking at? How reliable it is? And then how can you measure those signs in mice?
Yeah, in mice, obviously, you don't do the questionnaire. in humans, right? I mean, that's the thing. If you can't answer the questions and you get a score of 0, it pretty much means that you have become incapable of conversation. And these guys, they did not sign up for the study. No, they were signed up by their caretakers because they had no ability to speak at that point. So that's how large these effects are that we're talking about. And that's why figure 2 is seemingly so strong, because all the people with plaque clearance have MMSE 0, and that means they're in severe trouble. But there was one more question about mice. Oh yeah, how is it measured in mice? Right. So in mice you have similar types of questions, except of course not using English language, but you have remembering the way out of a maze. But the questions in the human MMSE try to mimic that. They're in Wikipedia. You should all take it. It's fun.
Okay. Oh, so the MMSE is the name of the test, right?
Yes. The minimal memory.
I'm afraid to take it.
Yeah, I'm afraid to take it.
Thinking the same thing.
You're afraid to take it, that means you already have it.
Maybe.
You're a negative 10.
So I'm a bit curious about the test they do with mice, and then determine that kind of what he was saying, that it's indicative of basically like a mouse model of Alzheimer's. How, if you're not objectively measuring the same thing in a mouse as you were in human, how do you make the leap that what works in a mouse might work in a human for treating Alzheimer's, or however you would measure dementia in a mouse?
Yeah, well, how do you make the leap? Well, you're a pharma company. You're running out of patents. You need new drugs. You have to make some bet, right? And if mouse results is all we have to go by, But people know that these are very bad bets, that, you know, most things that work in mice end up not working in people. That's simply what the numbers are. So surviving farmers simply try enough stuff to be lucky. Or if they don't have enough stuff to try, then we see only the lucky survivors. But it's very common that you bet on something in mice and in people it just doesn't hold. Especially if your mouse model is transparently engineered to be vulnerable to your therapy, right? If we're making the mouse by overexpressing Aβ, and that's bad for the mouse, and then we're removing the Aβ, the mouse is fixed, right? Because that model, it doesn't capture whatever the true cause in people might be.
Yeah, I guess I'm curious. So, you know, like in a mouse model, would something you would measure, as was mentioned before, maybe be like, you know, how a rat or a mouse would perform in a maze? Would that be more maybe spatial memory and something maybe neurologically you could measure more objectively? And then could you also do that in a person instead of using the MMSE test? So it's more, they kind of more are closer related together, if that makes any sense.
There is a problem even in that approach, because the way the mice remember spatial relationship is completely different from the way humans remember it. The mice has, I think I read somewhere, very specialized part of the brain that works on recognizes spatial relationships that we don't have. We actually have much more complex, multi-level, system in our brain. So you can't even compare that, even if you give the same test.
Yeah, completely agree. All this is making mice into very poor models. But, you got to do something. And, what else are you going to do?
And I guess monkeys is too expensive, too slow.
Mm-hmm. I mean, you have to start somewhere. If it helps the cognitive ability, whether it's remembering a maze or how fast I don't know if this is for Alzheimer's specifically, but there are tests in neurology where they will look at the pain reflex, like how quickly does the mouse pull its little paw away when you cause it pain, or how well does it swim? If you put it into water and it has a particular place to swim to, that it has to swim to get out, how quickly does it get to that little stool in order to get out? I mean, you have to start somewhere. I mean, those also require brain function. Maybe it's not the same type of brain function that humans have, but you can go through these assays in a couple months instead of like with monkeys that would take how long? Like years.
Okay.
So that would be my
So my point is, like, the comparisons are so rough, and the group number, like cohort number in this article is so small, and other article like 80 people is nothing, is so non-representative that I don't even know how they make any kind of conclusions here, to be honest.
Yeah, I completely agree with you. I don't think I can take anything away from this article with regard to whether or not this is a viable therapy. I guess I had another question though. Have they tried repeat dosing? And also, this is just like a question to the group, more of an ethical question. Like, if you found out that you had a genetic predisposition for developing Alzheimer's, like, how early on before you develop symptoms would you be willing to enter a clinical trial, like get a vaccination for something like this? Because that is where this is all headed. I mean, the reality is when you start developing symptoms for many things, it's oftentimes too late, when the symptoms are so severe. And so that means that treatments should start earlier. So how early is too early ethically? And I kind of just want to get people's opinion on that.
Are there legal limitations?
I mean, we vaccinate babies against their will, right? I mean, with things that are
proven vaccines, not with experimental vaccines.
Yes.
We don't vaccinate babies with experimental vaccines.
Yeah, that's my question. Is it legal to vaccinate someone who doesn't express any signs of, disease, and there is no clear proven causal effect? Is it legal to even, sign somebody in that study, even if they give consent?
So, I mean, legal is a question, right? What you got to do is consult with the Food and Drug Administration about your plans to do that. And then you got to discuss it with their scientists, right? And legal is whatever the outcome of that negotiation is going to be after, an excruciatingly long discussion of all the details of your case.
So this is coming up. There is no law that forbids you from signing in people in the experiment that can, kill them potentially, right?
No, there is no law. The law only says you got to talk to FDA scientists about it and get their permission.
I think in response to, is it Nina's question?
Mm-hmm. Yeah.
Like for me personally, if I had a genetic test that said I was very predisposed to Alzheimer's or oh wait, sorry, is that me? Oh. I think like before I went into it, since I think genetic tests usually show more predisposition than guarantee, depending. I would want to know, I think, a little bit what I might be getting into as far as long-term side effects, but then that's something you wouldn't know until you first did experiments.
Yeah, you're definitely asking too much there.
Encephalitis and death. That's the most.
For me personally, I don't think I would go, even if I knew about the predisposition, I don't think I would go in for something like this until I detected the earliest symptoms. And there's some very innocuous ones that you can find, like accusing people of stealing things from you. Like, I took care of my grandmother for like 7 years. Me and my mother were her primary caregivers. Like that's the way it starts. Like you can probably detect it before it gets to the irrevocable stage. If you're really keen on like what those subtle markers are for like just starting to happen. So that would be that would be when I check myself in.
So what about biomarkers? There have been some really useful biomarkers out lately. There was an article that came across my newsfeed a few weeks ago about a biomarker for Alzheimer's that was correlated with progression that allowed you to tell several years prior to the first psychological symptoms. Would you start treating those biomarker tests as a reliable indication that you should probably get vaccinated?
I think for vaccines, it really comes down to long-term safety. And just going back to what I said earlier about vaccines, a vaccine is meant to last pretty much all your life, or if you don't get booster shots, maybe, once a decade or so. Nobody's really tested long-term safety of any vaccine. You test short-term safety, and you don't know what the long-term ramifications are of having these, divided B cells in your body for the next 80 years of your life. You just test, is this vaccine going to have any acceptable death rate, for the whatever we're treating against, depending on what sort of vaccine you're using, an attenuated virus or a piece of DNA. So it all comes down to social acceptability and then FDA approval. So I don't think anything's going to be 100% completely safe. It's just a trade-off between what if you look at statistically what if you have these biomarkers, if you have this if you have a what's the marker for the SNP, the APO, APOE, right? So it's very statistically high correlation. Right, and then if you have a vaccine that has a low threshold of any kind of severe side effect, then it's green light go, right? There's really nothing else you can do other than do a 100-year study. There's just no.
Well, I think the initial question was, before we have those tried and true, it was like, how soon would you check yourself into one of these kind of highly experimental,
to me, it depends on the side effects. If the side effects are zero and the potential of getting treatment.
So the side effects are unknown unknowns at this point.
Right, but the side effects from this right now don't show that, oh my God, it's a statistically higher chance that you're going to drop dead.
So for one of the, I think this paper mentions it, but it wasn't this paper. Like some of the people that tried the drug got meningoencephalitis and died. So that's weird side effect.
Yeah, it's a stage 2 of the same research. 6% of people got meningitis, encephalitis, and they canceled the stage 2.
Right, but what's meningoencephalitis?
It was the immune response due to the antibodies.
Right, but how soon did that happen after they got like an injection? Does it happen like a year later?
I think it was enough. Like they aborted like
immediately during injection? Like, no, seriously. I mean,
but another study used slightly different formulation and they didn't have any severe reaction. The paper I sent, they actually did 80 people and like 60-something got it shots, and they didn't get anybody with the same reaction.
I guess for me, it would basically come down to just basically, acute toxicity, right? If there is no, I can't tell long-term anything. If I know for sure that there's a 99% probability or 80%, even 75% that these tests, they're stating that I'm going to have early onset Alzheimer's and that this vaccine has got no acute toxicity, then yeah, of course I'm going to take it. I mean, that's the best you can do. I mean, you can't really go by anything else.
A question on that. John, you were asking kind of, what can we kind of derive out of this paper? One of the questions I had that's related to the safety of the vaccine is, do we know that the cause of death in these cases isn't the vaccine? In other words, maybe the no plaques is the sign of that mild dementia, but somehow the vaccine is kind of taking a proxy for that, and you're suffering the dementia symptoms from the vaccine, not from plaques.
Yeah, that's an interesting one, and that's also in the discussion section, of course. So I guess the closest we can come to answering that is to look at the placebo, right? If the placebo is a sugar pill, then what you say is very true. But if the placebo is a mock vaccine, then we can have some statements about the mock vaccine possibly not causing that, right? And the placebo in this study was, in fact, a mock vaccine. It was adjuvant only.
What's the adjuvant? Sorry.
Then it would be specific to the Aβ sequence that you're vaccinating against, right? And that could still be that, dementia is not just caused by the adjuvant. It's not caused by the adjuvant alone, but is caused by adjuvant plus Aβ. And there's no experiment possible to resolve that.
What is the adjuvant, by the way? Because I know what an adjuvant in general does. It boosts the immune response for a particular, and they end up being all sorts of weird things like oils, right? Like what is this adjuvant that they're using here?
QS21.
QS21.
Purified plant extract.
Purified plant extract. That's the most general thing I believe. It's like cement. Purified plant extract. That's
purified. It's been tested as an adjuvant, according to it.
Yeah. Soap bark tree.
Soap bark tree. Okay.
Yeah. A soluble, tried, clean. Yeah, you can read this. Okay.
Saponins. So they're like soap-like molecules. So they're injecting you with soap.
Yeah, but aluminum for most vaccines, right? So.
Yeah. Yeah. The function is, like you say, is a dangerous signal, right? That alerts us to the peptide.
So then, John, that doesn't really sound like that's kind of a mock vaccine. Then that sounds pretty different if you're just getting the adjuvant.
Yeah, right. So it's possible to have.
You almost need to give the vaccine to some healthy volunteer here and see what happens to them.
That would be really interesting too. You're volunteering. But, I mean, this is a commonly used adjuvant, right? That does get used in other vaccines. And in those, we don't end up with end-stage dementia.
Right.
So it would be some Aβ specific effect that you'd be looking at, which we can't rule out. You never know what goes on once you're vaccinating against brain peptides. Aβ is part, of course, of the amyloid precursor protein, which has a physiological function, right? What happens if you target cells expressing that protein? That could easily contribute to the neurological problems that these patients are having. So I think this is a good question that's really not resolved and probably not resolvable.
I have a question about.
Oh, sorry. Yeah, you go ahead.
I was just going to have a question about some of the histology slides, and you'll have to forgive me, I'm not too familiar with staining with histology, but would it be something similar to where you would have an antibody and a fluorophore, and your antibody would bind to whatever protein you're interested in the tissue, and you would see those black dots?
Okay, I have to take a look at the methods section to get that definitively. Let's look at that.
I guess it's a two-part question. I have a follow-up to that.
So guys, this is Ryan. I don't know if
yeah.
So one thing that will actually yes. So hi from Counterculture Labs, who are actually live here. So one thing that the FDA, I mean, the challenge with using healthy normal volunteers, and I've worked with the FDA, and particularly in the Alzheimer's and CNS space, that they typically won't allow higher risk projects like this in healthy normal volunteers, but that would be a great question to ask. What happens if you were to vaccinate a healthy normal volunteer? Do they get sick? And who knows? Right? So that's something, unfortunately, it's a test, as John was saying, we probably won't be able to run. The FDA won't approve. Now, if some biohacker decides to vaccinate himself,
I just think for these types of treatments, we're not really going to get an answer unless we start earlier on.
Yeah, but the problem is if you start too early, then you will not be able to publish in another 20 years.
It's got to be like Fringe style, you know. People.
Guys,
Guys, sitting on a sofa. You're sitting too far from your microphone. We barely can hear you.
Let me try and move up a little bit. So, guys, are you familiar with the Genentech study?
Nope.
Yeah, familiar with the Genentech study?
Are you? I'm not familiar with it.
Okay, so Genentech, and John may be able to jump in on this. John, they've just recently started on a population in Colombia, right? Yeah. That's Jamie actually knows a little bit about it as well. I don't know too much about it other than the fact that they're going for familial Alzheimer's, and they're going for a much younger population. So almost a preventative study, which will be very, very interesting to see what the result comes out with.
Mm-hmm.
So that's something to look up as well. Cool. I don't think they've released any results just yet. But so
is that based on people having having mutations? Yeah. Okay.
It's genetic.
Yeah.
And is that testing the same vaccine then?
No, they're actually. I don't think it was the same vaccine. I think it's something that Genentech has developed, which is separate from the Elan and J&J vaccine.
But it targets it kind of in the same way?
I believe it's I think they may be looking at Aβ, but I may be wrong. So.
I want to say it's a monoclonal antibody.
It's a monoclonal antibody. I think you're right. Yeah. Yeah. So Jamie thinks it's a monoclonal. I think he's.
Okay, so I just want to come back to one other objection that went unanswered so far, which was about numbers of patients, right? Somebody just said there were only 80 patients in the study. Yeah. But if I go back to figure 2, I really count 8 patients.
Yeah.
So that's the question, is figure 2 cherry-picked? Well, the answer to that is yes, absolutely. Nothing gets published that isn't cherry-picked. But how badly is figure 2 cherry-picked?
Yeah, you see there is an Asian mathematics anecdote I keep bringing up. If I ate a chicken, you ate nothing, an average weight of half a chicken. You know, that's the kind of stuff that this article produces.
So, I mean, in other words, the question is, if we have Alzheimer's patients and we slice their brain, can we find a several square millimeter slice where there would naturally be no Aβ occurring, right? Is it even possible to find a slice, or does all of the Alzheimer brain look like the unimmunized patient? Well, we kind of have that in the figure there. I mean, look at the unimmunized patient, right? They have a zone there with lots of dots, but they also have a really big zone there with no dots.
Yep. Yeah. And we have one that immunized with a lot of dots. So, like, you know.
So, you know, I think you have a very good point there in saying that, you know, with 8 patients, it is entirely possible that we're cherry-picking the whole thing out of noise. Right, that is a good question, and I think
this
very much
has more variables than patients. You know that
actually correct. Degrees of freedom.
Yeah, more degrees of freedom than patients. You know, there is not even enough patients for 1 degree of freedom. So, you know, that to me makes all this study conclusion, except the one that says that this vaccine is not safe. And it seems that's what the consensus is among the scientists who do that, because the study I sent later on that was done by Novartis in 2012, published by Novartis in 2012, that's what they say. They come up with completely new formulation because the previous vaccine was too bad. So they come up with new one and just ran the phase 1, the clinical trial, where they didn't have any, previous adverse effects, but they don't publish any results at all. They just, they also around 80 people, but they just try to check if it's not as horrible as the previous one, and that's it. And this study, it seems to me somebody's trying to make a PhD out of nothing. If you can correct me, because I'm not a biologist, but from my eye, it's just trying to suck a PhD out of a finger.
Unfortunately, this is beyond PhD.
What do you mean beyond?
This is like a company's survival.
Okay, so then somebody tries to impress shareholders with something, but
Well, let's see about it. Okay, so now we're willing to consider the world where figure 2 is simply people pointing the camera at enough slices until they find one, proving that there are patients with no beta amyloid who are severely demented, right? So if we're willing to consider that world, maybe we should think about whether the company has a financial incentive to consider that world. So who can think of an argument of how somebody might profit from the claims we're drawing from figure 2?
It's easy. As a CEO of company, you depend on your investors. So if you can convince them to keep investing in your company for longer, you increase your chance to find a real treatment, or a real blockbuster. Because if you don't convince them that you have at least something, they're just going to pull the plug on you, and that's it.
Okay, but so what Elan is doing, right, they followed up on this with an improved vaccine, which is a monoclonal antibody, a passive vaccine, right?
Yes.
Also targeting beta amyloid.
Yeah.
Now, the problem is this paper shows, successfully targeting beta amyloid does not help. If that's wrong, why are we developing a better therapy that's also trying to target beta amyloid?
That study doesn't show that. It doesn't show anything except that it has an adverse effect.
No, it doesn't show that. It claims that, right? But if that's a false claim, then, how does it make the shareholder continue to invest in beta amyloid targeting therapies?
No, it makes it look like it has a potential for that. That's all it says. And
No. It would say, if it was true, it would say that even if we successfully target beta amyloid, we're still not going to have a working drug. Right?
No, it could work in drug if we can, but it says you can have an inflammatory effect of vaccine that negate the good part of the same vaccine.
That's really going out on a limb there.
Yeah, it's in the end in discussion. So it just opens the door and say, we didn't screw it up completely. Here's possibility. It's cleaned up, and probably something else, didn't let it work. But we're on the right track. Give us another billion and another 5 or 10 years, and we probably come up with something.
Oh, they did. Not just 1 billion.
John, did you say that the kind of the competition in this space decreased after these results? People stopped doing development of these drugs?
Yeah, we've had since then efficacy results announced from several companies, Eli Lilly and Elan, from monoclonal antibodies targeting beta amyloid, and all of those were completely inefficacious. And since then, the pipelines have been rather dry. And there were also small molecules trying to inhibit the enzymes that produce this thing, and all of them were completely inefficacious. But we don't necessarily know what happened to the beta amyloid load with many of these drugs. We just know that they were inefficacious from a disease viewpoint.
Mm-hmm.
So I have a question just outside this article. Is there any prospect of treatment Alzheimer? Does anyone have a better idea than treating amyloid?
All right.
Right now.
I think that's what John wanted us to talk about. Are beta amyloid proteins the right target?
No, but if we say that it's not right, does anyone work on something else? Or
would they like discussion about tau inclusions and other things, right?
Yeah, Ryan would probably be the best person to catch that. Ryan, are you there?
Hey, Ryan, question for you. Hey, John.
Hi, guys.
Ryan, are you there? Yeah.
I was sneezing. I had a little cat allergy, so.
Okay, Ryan, the question was, what other approaches are in the pipeline not targeting beta amyloid to Alzheimer's disease?
We can hear you. Sorry, you're too far.
Okay. Yeah. So there. Can you guys hear me now?
Yep.
Yeah. Okay. So there are a couple of alternatives to the A beta theory. Actually, there have been some interesting stuff. So some companies are actually going after subunits. So there's some companies that have a hypothesis around the subunits involved in A beta, which are not, A beta is not just A beta. A beta has different subunits, and that's actually really important. So there may be a nuanced difference between different types of A beta. So some may have nothing to do with Alzheimer's, and some may promote it. So even within A beta, there's differences. There's tau, which again is another hypothesis that John isn't a fan of, right, John? And also, there's some correlation, but again, it's a debatable hypothesis. It's the minority hypothesis still.
Okay, sorry, but Ryan, the question was drugs in the pipeline.
Oh, sorry.
That's why I called you, right? It's a pipeline question.
Oh, okay. So I'm wrapped up in a bunch of NDAs and things.
Well, public ones. That's okay.
But let's just say the dominant therapeutics right now are still targeting A beta. There are a handful in tau, and there are a few other mechanisms that have been tried and failed. So there was a company called Medivation. I don't know if you guys have heard of Medivation. This is one of the public ones. Early on, I was involved with testing Medivation's. It was actually an antihistamine from Russia, which was actually being used, and they thought there was a theory around it actually being able to reduce Alzheimer's or reduce neuronal cell death. Turned out it was wrong, and it failed in like a big phase 2 or a late phase 3, an early phase 3. So right now there really isn't a lot in development. There's still a lot of what I think early development has been either treating the symptoms or the dominant hypothesis. There is, and now this is actually very interesting. Some people are actually trying to use metformin to treat Alzheimer's.
It's used in diabetes.
Oh, okay. Yeah, used in diabetes, exactly. So that's to do with the hypothesis around Alzheimer's and kind of a pre-diabetes or type 2 diabetes link. It's kind of a diabetes of the brain. It's an interesting hypothesis.
Okay.
Why would that be? Do they think it's elevated glucose somehow oxidatively damages the brain? Is it,
maybe.
What would metformin even do?
Metformin is an interesting drug. It may actually be linked with longevity as well. So I'm not sure. I'm not sure exactly. That's like a fringe hypothesis that I haven't been actively involved in any of those clinical studies, but I know that there is that interest. And I haven't looked at the most recent. There's probably some stuff in earlier phases, probably neuroprotectants and anti-inflammatories that are being looked at as well as a broader class of drugs. There's actually one drug which I've been involved with, which is called J147, which is being tested at the Salk Institute. It's actually a chemical modification of curcumin, and it's actually been curcumin for the anti-inflammatory effects. And the molecule was actually changed, so it was also neuroprotective as well. It showed some promise in early mouse models, but that's it. So it hasn't even touched humans. So this is a tough space.
Thank you for giving hope.
Yes. Yeah.
Okay, so we had anti-inflammatory and antioxidant, and the third thing I want to add to that is metals. So that's chelation therapy, right? That's Prana Biotechnology is the most advanced compound in that class. But none of this is in phase 3, so yeah, hope's what we got.
Hope is important, yeah.
Good, thank you.
You're welcome.
What I'd like to see is the word ApoE was mentioned several times around here, right? That is, of course, the gene that confers natural protection from Alzheimer's disease if you have the right version, right?
Do you have any updates for us on ApoE?
Well, no, all I have is hope. what I'll say is, you know, since we know that this thing confers natural protection, I would like to see therapies based on that, But you know,
what does ApoE code for again? What does it do?
ApoE is the carrier protein for the HDL, the good cholesterol particle.
Right. Oh, cholesterol.
And that points us to a cholesterol hypothesis of Alzheimer's disease.
Right.
Who has ever heard of that thing?
Yeah, exactly. Yeah.
That ApoE is an interesting one personally for me. I had my family do a 23andMe test about 2 years ago. Was all hyped on 23andMe results and thought it would be fun to get my family to do it. And I turned out to be an ApoE4 heterozygote. My sister turned out to be an ApoE4 homozygote. And some of the stuff around the family talking about, well, is it really good to know these results and what can we do, have been really interesting discussions. I think the summary is that, yes, everybody in the family now thinks it's a good thing to know, except my father, who decided not to get tested. And with my sister and I having both at least one of these, he's now aware he's at least a heterozygote against his really wishes to know about that.
Okay, so who has ideas for ApoE-based therapies?
Vegan diet.
Yeah. Ben had an excellent suggestion. He suggested the vegan diet. I too thought.
You need a small compound that can overexpress ApoE. Maybe something that targets. Maybe there's an ApoE-specific transcriptional activator.
But if you have the bad version of it, then you'd be overexpressing the bad version.
Well, you still have a large market to, for people who have the good version.
Sounds like a
protective effect. Or downregulate the bad version. I don't know.
Mm-hmm.
If you have the bad version.
So John, what about gene therapy with ApoE, with the good version?
Yeah, I don't think anyone's tried it.
Okay. So that's interesting.
Is there anything known that would is there any other drugs that are known to affect? Let's say you have the bad version of ApoE, and I don't know exactly what the bad version encodes for. Is that mutation? Is there mutation in ApoE, or is there less levels of good what does this mutation do exactly?
There are 2 point mutations that change the amino acid sequence of the protein in 2 specific points. Those make the versions.
And Oliver, it's an HDL story, so that we do know that there's a drug, a very old drug called nicotinic acid,
right? That
obviously increases HDL.
Mm-hmm.
So that's a B vitamin.
So is there anything known out there that if you have the bad version will have a decreased level of ApoE?
Yeah, like you have E4 version associated with increased risk. E2 is rare but protective effect.
Like, is there some cross correlation with so let's say you measure levels of ApoE in the blood, and people are taking all these different drugs, and we just mentioned that metformin might have some effect on Alzheimer's. Nobody would have predicted that. There's some broad meta study being done to see, this group of 100 people have the mutant form of ApoE, but they have low levels of it. Oh, and they all seem to be taking a particular antihistamine. I don't know. Is that a particular direction?
I think that's a great idea. I have not heard of anyone ever looking at levels. The ApoE is traditionally a gene test, right? That doesn't tell you anything about what's happening right now.
Right.
Yeah, expression. Yeah, that's actually a great idea.
I'm curious what's the statistical correlation between ApoE and Alzheimer's?
It's very strong. If you have homozygous E4 and you're 85, you're pretty much guaranteed to have it. And if you look at Alzheimer patients and you type them, none of them has homozygous E2. It's that strong. It's 95% of the disease explained by that SNP.
I.e., John, it's protective, right? So if you're homozygous, it's protective.
But it depends on what you're homozygous for. 2 is protective, 4 is bad.
Okay.
Yeah, it's actually a combination, yes.
And 3.
So would somebody like say you made the E was the good one, and if you made that protein and injected it into somebody, would there be an immune response to that?
You make that protein, yeah, good one. That has been done for heart disease once, not for Alzheimer's though, which is too bad. The company that was able to make these proteins was called Esperion. And at some point, they were bought by Pfizer and shut down because they were endangering Lipitor sales because they were trying to develop in heart disease. But maybe they should have developed in Alzheimer's. Maybe the world would be really different now. Yeah, don't know.
Yeah, sometimes I guess I was curious about developing the E2 protein and then perhaps an enzyme for the E4 protein.
Yeah, the thing is, injecting the protein, that actually works in some diseases as opposed to gene therapy, right? The only problem with gene therapy is that we haven't really gotten it to work yet anywhere.
Yeah, right. They have one commercial use of gene therapy in Europe now.
Oh, really? What is that?
Give me a moment.
Okay. All right, well, that's good.
So for ApoE, we actually know that you can actually give that as a drug, and just orally, it'll get taken up fairly easily by the body. We have people at Livermore working with these little nanolipoprotein particles. ApoE actually forms like a little ring that you can fill up with a little patch of membrane, essentially. Turns out you can actually then load that membrane with various membrane-attached proteins and use that as a drug. And the whole particle actually gets very well tolerated by the body. So that is actually a pretty promising drug approach, potentially.
Interesting. Okay, cool. So if I may summarize our current conclusions is that our paper has a serious problem with potential cherry picking. And we shouldn't read too much into it. So are we willing to conclude that it's possible that Aβ remains a viable drug target? Are we willing to state that?
Maybe.
Yeah, I think.
Yeah, maybe.
What probability?
So yeah, maybe.
Type your probability in the chat. What probability Aβ is still a good drug target?
We should have that bar graph on like Who Wants to Be a Millionaire go up.
Yeah. Everybody type in their answer. I'll summarize the results later.
What about plus or minus 50? There you go. I think over here consensus. What's our consensus? I think 30. Counterculture Lab says 30. Which percentage?
Like the one that finished.
That's fine.
Yeah, that's the question that makes medics think about the bronze statistics. Don't even ask that question. Because it's like a question, what the probability to see a dinosaur on the street, and people. The same as here, no.
But have we ever done this type of study? Maybe picking a drug by voting actually works. Has anybody actually tried that? All in favor of this drug? Okay. Have we tried that study? No, we haven't.
But my opinion is that beta amyloid was implicated to be in a gang of things that contribute to Alzheimer's. So it's good that it's an good investigator. If you've got one guy, it gives you a better chance to get the whole gang. We just don't know if that guy is the main guy. So I think they still have to pursue that direction. Because it seems he was caught on camera, so probably he was a driver, but he knows who killed
Hollywood land.
Yeah. But it's easier to talk that way.
What kind of gun did he have?
So I think they should research it. He probably will not be end up the one who causes it, but who knows?
What kind of car was he driving?
Yeah.
He takes his filthy black.
Yeah, he's probably like this black guy, but it's probably a blonde black guy, stuff like that.
If you're still interested in what that gene therapy was in Europe, it was called Glybera. Glybera.
Got it. I will check that out. Thank you. Okay, good. So as far as this paper is concerned, it seems like the overall consensus is that it may reduce the chances that, you know, A beta is important by a little bit. We got something in the low 30% on average so far. That A beta may still remain a viable drug target, so that means 70% say we should abandon it, 30%-ish say we might keep it. You know, when drugs are worth billions and billions of dollars, might still be worth a gamble then. And with that, I'm done, and I'd like to hand the mic back to Nina. Thanks, everybody.
That was awesome. I also think we came up with some project ideas.
Yeah.
Yeah.
Now we just need a space to do them in. We need like we need a vivarium. That's what we need. We need an animal lab.
Well, let's put together some proposals, name a company, and go to Calico, the new
Google venture.
The new Google venture that wants to fund Alzheimer's research and longevity research in general. And let's all sit on the board of directors, and that's it.
Done.
In a week, it's done.
It's actually not a bad idea.
No, no.
Yeah.
You want to summarize? You want to summarize what the project ideas are?
Yeah, sure. So project ideas were gene therapy ideas, right? So we could target the SNP, or we could replace with a wild type copy, right? So those are various forms of therapy.
Can you explain to the non-biologists here how you're going to change the SNP in the living brain?
Yeah, that's hard to do.
Yeah, how are you going to do that?
Okay, so first thing is the ApoE, right? It largely, most of it, does not come from the neurons, but it comes from the microglia, which is the immune system of the brain that's going in there and trying to clean up cholesterol deposits or even A beta plaques. And then it is synthesizing that and packaging it onto. Now, the neurons contribute to that too. They make some amount of the A beta, but the microglia makes most of it.
So I have another question. Can we just synthesize ApoE in yeast and inject in the brain?
The E2 variant? Yeah.
Will it be faster and simpler way to do that?
Straight to the forehead?
In theory, you can. So the only problem with your question is the just, right? It's not easy, but it's possible in principle. Like the issue is that the ApoE is part of a protein lipid particle, right? And that has nanostructure and stuff that's really hard to make. And it's not very soluble by itself. Its purpose is kind of to solubilize that lipid particle, right? Yeah. So this is easy when you're a cell and you're making everything at the same time. But it's hard when you're doing it. But it has to come off.
Oliver had also mentioned some kind of meta study looking at
like a drug.
Levels.
Like I don't know what metformin does.
Oh, right.
Something out there like a soluble molecule that might affect, either boosting ApoE levels if they're the good ApoE, or bringing ApoE levels down in a person who has the bad ApoE. I don't know what that correlation would be, but you could start off on the study maybe. I mean, people are taking all sorts of potions out there. If you can just look at various ApoE levels and correlate that with people that are having break them up into cohorts, like people getting Alzheimer's, people that are getting early onset Alzheimer's that have this mutation for it. Are there less of them getting that if they're taking a certain drug? I mean, I think the problem is probably there's studies that are being done where people are trying to post their medical history online for researchers to look through. Correct me if I'm wrong. The problem is that this would require a lot of people. It'd be easy to do if you have that data set, to sift through it. But a lot of this data is binned, so it's not available. There's not, we just don't know who's taking what drug and what their ApoE levels are. They're walking around out there. It'd be nice to have that data.
Well, that's stuff like the website Lyle Dennis was trying to set up, where instead of social.
Well, yeah, there's a website out there where I forgot the name of it. There's a guy at a TED conference, at a global conference, who proposed it, where it's basically you're uploading all of your medical history and what you would, so to make this data available to anybody to look through and to do these types of
genome.
No, it's nothing to do with genomes. You can put genomic data as well, but you can put your blood, your blood tests up. You can put what drugs you're taking, so that you might be, you might make interesting correlations with things like, like who would have known that metformin might have some effect on Alzheimer's a priori. I mean, you're right, the two don't go together. I mean, having this data set is completely unbiased. You're making a correlation without any presuppositions as to what drug is having an effect on what, and you're just mining that data.
I know one person that has at least a site like that called HealthKeep is Lyle Dennis, who I think runs like radicallifeextension.com or something like that. But his goal was to do that. He launched it like a year ago. I don't know if he has a viable data set yet, but the purpose of it was just what you're talking about, was to have people sort of anonymously contribute their whole medical history to be used for meta-analysis.
Mm-hmm.
So.
Hey guys, before we run out of things to chat about, I'd just like to mention that we have the people here from DIY Sect. So it's the two that are doing a little DIY bio documentary. I think I'm blocking. There we go.
Hey, hey guys.
They would love to shoot a little video of us chatting, I think. I don't know if you want people to introduce themselves or...
No, just keep it natural. Just keep doing what you guys are doing. We just want to get a little...
So just cut off all the politically incorrect comments made by me, okay?
You're already on it. You're already going to be on a YouTube channel. We're going to have to edit you out.
You got to edit him out. He's going to be hunted by Alon's assassins.
True.
Cool. All right. Well, great. Keep going. Do your thing.
Okay, so
right,
how many people?
Set up and just shoot a little while we're chatting.
So how many people have submitted their samples for 23andMe on here?
Yeah, I have all my family on 23andMe, and I'm looking at ApoE.
I have to go back and look now. I don't remember. I didn't know a little quantified self-study. Like, I'd be interested in changing my diet or something, I mean, what are things that are not too risky that we could really actively participate in?
I'll look this up. There was some paper that was published recently, I was mentioning to Keith, where there was like some nutraceutical supplements that
like omega fatty acids.
I don't want to even say them because I don't remember them, but they're basically, it was either Alzheimer's or dementia, but it seemed to have an incredibly strong correlation, protective effect for people taking these substances, and these seem to be on the surface benign substances, things you would normally take as supplements anyway. It was a cocktail of 2 or 3 of them. So I have to find that initial study, but it came out recently. I don't know if anybody knows what I'm talking about. If somebody saw this, it came out within the past year.
I don't know the particular study you're talking about, but I saw an interesting AAAS talk about a year and a half ago with the researcher giving the talk attributing a strong protective effect to coffee. And so he was drinking 3 cups of coffee a day now as one of the best effects against Alzheimer's.
Don't be wired and confused.
Exercise is also very protective as well.
Oh, yeah.
Yeah.
So I don't know if you caught that.
Yeah, especially physical exercise. We found like it's even better than mental exercise, whatever being that. That's what we're finding is that actual physical exercise is more effective than doing process.
Can you guys hear that?
No.
Oh, okay.
Sorry. I guess also exercise is neuroprotective. When I was working at the MAC at UCSF, we found that exercise was more neuroprotective than doing any crosswords or any of the medications that people, I think the medications are probably the medications.
Flat out regulation.
So then that makes more sense taking more caffeine would have a protective effect because you're more Energetic.
Yeah, but you see, again, getting back to how you diagnose, Alzheimer, people who are predisposed to exercise, they can have different brains, they can have different genes that,
Well, sure, or even if some of the tests on the MMSE scale wouldn't have to do with, well, that I think was questionnaires, but if there's any sort of physical assessment of, can you walk quickly or do something.
I know, it's very hard to make correlation and causation.
Yes, that's what I'm saying. Like, the people who are more active, they can just die earlier because they got in more accidents, so their statistical amount of getting Alzheimer can be lower.
No, I'm sure they correlate for that. I'm sure they, I'm sure it's an age. Obviously, they're not.
How you correlate?
I mean, you have to take somebody at the same age group. Clearly, they can't. They're not going to count somebody who dropped dead at age 20 because of a bus accident in the study.
Yeah, that's the problem. Anyway, so one more thing. I set up a mailing list for this journal club and and included everyone I found in Oliver's email. So if you got a message that you were added to mailing list, there is an address there to which you can send mail, and it will distribute it to everyone. So nobody got lost if you distribute article or some comments on the etc. If you're not on mailing list, send your mail to Oliver or to me, and I can add you. And Nina, I can make you an admin of that mailing list too, so you can add people. And
awesome. I didn't get an email, so.
I didn't either.
No, you.
I don't think you sent one out.
What?
We didn't get emails. I don't think. Did anyone else get one?
No.
No, I added it, and the shoot sent out. Okay, I will check out.
Okay.
Yeah, I will figure. Did you get a mail from me with 2 articles?
No.
Yeah, I actually got that email.
Oh, you did? I didn't get that.
Okay, so you're.
I did not either.
You were not in list. So, Kenina, I will add you to the distribution list, and you will be able to add other people on other, costs to that list.
A bunch of the West Coasters weren't on it, so I think we're looking forward to joining.
I added everyone who was on Oliver's mailing list, in his.
No, everybody that was on the mailing list, probably Nina wasn't probably on. Never mind.
Okay, we'll figure out how to add people, so we will have a more convenient way to communicate other than Oliver not having everyone in his mailing list.
Yeah, somehow it just.
Because you usually accept the blame, so
that's true. I'm a good sport. Yeah.
Anyway, so if we are, are we serious about potentially looking at, setting up our APOE2 protein factory or whatever? Or what would be the?
I can use some synthetic biology to make some of that.
Yeah. Dealing with animals is going to be the hard part, though.
Oh, absolutely.
And if you guys can do it, I can talk to Big Pharma and biotech. I know some of the research heads over there.
All right. Let's get on this.
You can use me as a guinea pig.
There we go.
Yeah.
That's awesome.
Yeah, I used to do APOE genotyping, actually.
So we need another bee.
So Ben's volunteered himself. He's an APOE2 mutant.
Wow.
Yeah.
It's going to be like, Wolverine. Let's see if
yeah.
Okay, Ryan, get his bone marrow. Now.
We'll get a little big. We'll go.
So if you're talking seriously, the place where you can find volunteers, 23andMe, you go and find the people with E4 allele for ApoE, who has both allele E4, so they have like 9 times higher probability of getting Alzheimer's disease. So here's your group from which you can fish out people. And there are hundreds of thousands of people on it, and there are forums, and people actively participate.
We'll just show up at their door in like black suits and sunglasses.
So.
Okay.
No, the black suits are for getting the E2 carriers.
Yeah.
The E4 carriers are going to volunteer.
Sure, that's true.
But you need to get the corrected cells from somewhere, right?
Okay, so I'll work on setting up the SWAT team. Nina's got the synthetic bio covered, and we got the donor. All right, so we're in good shape.
Easy.
But
we could package some genes and some viruses.
And then we could go and talk to Big Pharma and see if they'll fund it. Right?
Yeah.
So does anybody know what the ApoE4 mutation actually does to the protein? what structural change and functional change it causes?
Yeah, I think that's what Oliver had asked.
Yeah, but I don't know if yeah, I did ask that, but I don't know what it actually does in the protein, right? I mean, you're asking about structural.
Can you pull up? Is it possible to pull up the variants of the proteins in the protein data bank?
Yeah, the structural changes are easy, like you say, absolutely. The structural changes have been characterized, but what it means in the context of a body or of Alzheimer's disease, we don't have any idea.
Well,
I might.
It was implicated in the good cholesterol. Like, does it change the way it interacts with that, or like has that been any attempt to characterize what those ApoE mutations do as far as the interaction with the cholesterol?
Yeah, that's heart disease research that has characterized that in a fair bit, right? The good cholesterol is good because it's cholesterol on the way out. So if you have a lot of it, you're getting rid of a lot of cholesterol. That's what makes it.
But this mutant, is it less efficient at doing that? Like, have there been in vitro studies or even in vivo studies that showed, like, what does the mutation confer? Or
no, the weird thing is the E4 does not by itself predict susceptibility to heart disease, only to Alzheimer's. And we don't know what the role of its cargo is in Alzheimer's. We know it only in heart disease.
Yeah. It's pretty important to know that.
Yeah.
Just from actually from the Wikipedia page, it's a very reliable source. Apparently, ApoE enhances proteolytic breakdown of the peptides that form these aggregates. And if you have the ApoE4 mutant, it cannot catalyze that reaction as well. So that's when you get the buildup of these aggregates when you can't break them down. So there are two.
Is this directly interacting with those aggregates, or is that a long chain of events in between?
I don't know at this moment.
That have shown that Aβ gets loaded onto good cholesterol particles and sent to the liver for excretion. But, you know, there's the truth, and then there's whole truth, and then there's nothing but the truth, right? So the question is, you know, you can, in the body so much is going on, you can always show stuff like that. But, you know, is it relevant to driving the disease? That's the hard question that we can never really answer.
Out of curiosity, does anybody know the ApoE protein? Does it cross the blood-brain barrier? Or is it primarily made in the brain when it comes to Alzheimer's?
I think it's too big to cross.
Yeah, it gets out, definitely. It gets made there, and it gets out and goes to the liver for excretion.
One thing we're looking at these nanolipoprotein particles is that they actually tend to fuse with the membrane of the cell. They have other ways of crossing barriers that normal particles would have.
Yeah, I believe they have address tags that tell the blood-brain barrier to actively transport it through.
ApoE, this whole complex, this actually can get through the blood-brain barrier. Like,
yeah, I think there's an enzymatic route for that. Let me look that up now. Wow, yeah, there are studies enrolling now to study whether the different forms of ApoE get through the brain barrier equally effectively. There's interesting stuff coming out there.
They scooped us.
I love this. We're sent to an army of scientists right now, which is pretty awesome. Right? Cool.
A wall full of the DIY bio people.
Exactly. Citizen scientists / real scientists doing science.
A lot of science.
Okay.
Yeah, Google, when I typed ApoE and brain, it's, crossing brain barrier, just added to, self-corrected my query automatically. There's a lot of people looking at that. So, should we end?
Yeah. So are we at the stage where we pick something else for next week?
Yes. Let's do it. This was awesome.
Or found a corporation.
Or found a corporation.
Yeah, I think, well, one, we should definitely start a company.
Now we realize the bar is quite low for the Lancet.
Yeah.
No, the bar is pedigree, right? That's the hard part to getting in the Lancet.
We need no statistics, so unfortunately we don't need your help.
Okay, so what should we talk about next time? We're in 2 weeks from now. Should we continue on?
Sorry, Lancet, if I ever decide to see it.
Should we continue on the brain? Or should we move to another topic? What do you guys think? What would you like to talk about? How do you guys feel about viruses?
Awesome. So,
Sounds interesting.
Yeah,
what kind of article we're talking about?
I don't know yet.
Regular viruses or those big mega viruses?
The Pandora virus.
Oh, the Pandora virus.
Is that the big one?
Yeah. Viruses can be the size of softballs on occasion.
So some of these Pandora viruses are actually fairly easy to get out of the environment because they're literally using an amoeba culture to go hunting for them in ponds and soil and whatever. So this is actually something you could do in a DIY setting. If you grow your own amoeba, you can try all kind of different samples from the environment and see if you can find some cool viruses that you could actually see under the microscope.
What about, speaking of amoebas, what about that amoeba that eats human brains? Can we come up with a therapy against that? Maybe a virus that infects Naegleria fowlaria. What is it called? Fowlaria naegleria?
Yeah.
That would be cool.
And then we accidentally kill ourselves in the process of work.
Let's go on a DIY expedition for brain eating amoebas.
Let's pass on that.
They're everywhere. They're easy to find.
I vote not to have my brain devoured.
Science magazine has an article about once in lifetime flu shot. It's viruses, it's vaccines, and it's interesting for everyone because everyone gets flu and everybody has, getting flu shots every year. So, what do you think about it?
Sounds good.
Sure.
Yeah, it's published September 13th. So,
I think this week in virology did an episode on that as well.
Yeah, I heard the news. That's why I found it. I was listening to podcasts and they were mentioning about it, so I found the article.
Do you guys want to do that?
Sure. Wait, does that mean you're volunteering to?
Oh no! I know very little about viruses. Probably it will be enough for 1 minute, and that's it. So somebody who understands all that stuff.
Send me the paper. Send me the paper.
I will send you a link. So because I don't even
Yeah,
have.
send me the link. Yeah.
Okay.
Okay, cool.
Are we good to go?
And if we don't like it, we can use distribution list, to bounce other ideas.
Awesome. Thank you so much for setting that up.
Okay.
Ben's also suggesting HPV as a backup. If anyone's interested, I could.
Was that human papilloma?
I can hear you guys again.
Yeah, HPV. Yeah.
Ben's suggesting HPV.
Okay.
What HPV? You have an article in mind?
No, just some experience.
He says he has some experience.
Okay.
No, not personal.
Phrase that if you want.
There's also some really exciting interactions between viruses and microbiome and the human immune system.
Yay, bacteriophage and the microbiome.
That actually sounds really interesting.
Not just bacteriophage, actually human pathogenic viruses, but their pathogenicity is modulated by the microbiome essentially.
Oh, I didn't know that. That sounds really interesting too. You
the crossover episode.
Wait, would you feel comfortable talking about this?
Not for next time.
Okay. Yeah. Okay.
The one after that.
Oh, the one after that. Okay. So Patrick will do the one for next time. Not the one for next time. The one for after that.
Sure. After Halloween, I'll volunteer for something.
Okay. Okay.
Perfect.
Yeah. Okay.
You know, one other thought. I wonder if we might get the host of TWIV actually to join us for some virus talk.
It'll be fun.
That would be awesome.
And, yeah. So if people don't know, this is This Week in Virology and This Week in Microbiology are some of my favorite podcasts. It's the same host, and he's really good at actually reading listeners' emails. So if you ever want to put in a plug for your DIY Bio Lab, you can probably get something in on the radio.
Yeah, it's Vincent Rankin-Yellow, right? Rankin-Yellow.
It's a really good podcast. It's essentially it's a little mini journal club among a bunch of experts in the field.
Very cool.
Okay. All right.
I have a question for all of you real quick. Next time I'd be interested in getting some more people from Portland involved in this. But I'm wondering, where do you find the conference number for these sessions?
Oh, Derek set up the conference number that I sent out, that Zoom link. That's a standing conference number, so we can use that every meeting.
And what do you send that out on? What do you guys primarily use to communicate on?
I was sending it out to the BioCurious listserv, Counterculture. I have a DIY Bio Idea Lab group, and just random people. But feel free. I mean, and then we are also trying to post it on the meetup pages for BioCurious.
I'm just looking, Nina, and I think although the intention was to have the link the same every week, I think I screwed up and did not set it as recurring. So I think that has to change one more time before it's going to be the same every time.
Ah, okay. Okay.
That's the whole idea behind mailing list, so we could use one point to distribute it. Nina, I have just added to your the mailing list. You can go to the Google group page and add other people. You're not made manager of that mailing list.
Awesome. I just got it. Thank you. So yeah, we'll add you to the list, and then you can add the Portland folks to that list, or you could give me the list of Portland contacts, and I can add them to the list as well.
All right, that sounds good. Should I just type my email address in the chat then?
Perfect. Yeah, I also have your email address from earlier today, so.
Oh, perfect. Great.
Okay.
I think we're good.
Okay. All right, awesome. Okay, I'll send out information soon with regard to next time's discussion.
Sounds good.
All right, thank you all. This is great. Thank you, John, so much. That was awesome. I learned a lot tonight. It was great. Yeah.
Thanks, John.
Thanks, John.
Thank you.
Thank you all for lending your brains to take this paper apart. That was really good.
All right, I'll see you all soon.
Bye.
Bye.
Bye.